overall goal of this technique is to study the supramolecular organization of photosynthetic membranes using cryo-scanning electron microscopy of freeze-fractured leaf tissue samples.
This method can help answer key questions in cell biology, such as the morphological characterization of cells and organelles, the distribution of membrane integral protein complexes, and localization of biominerals.
The main advantage of this technique is that freeze fracture is performed on leaf tissues, and so thylakoid membrane supramolecular organization is investigated within the native organelle and cellular context.
While this method is demonstrated here for a biological sample, it can also be applied to study soft matters such as gels, polymers, and drug delivery systems.
Generally, individuals new to these procedures should be aware that not all samples will be well frozen or properly fractured.
To carry out cryofixation of leaf tissues by high-pressure freezing, begin by using the corner of a razor blade to scratch the bottom of the 0.1 mm cavity of a 0.1 to 0.2 mm aluminum platelet. Prepare at least a dozen platelets for 6 samples. Then use absolute ethanol to wash the platelets and keep them in a clean dish. After preparing the high-pressure freezing machine, fill the samples box with liquid nitrogen. With a razor blade, cut a